Well, hello, everyone. I'm Michel, and it's a real pleasure to have you with us today. And thank you for taking the time to join this session. In this webinar, we will be hearing from Professor Roger Barker on stem cells and Parkinson's. How close are we? We would like to have the answer to that tonight. Roger, Roger Barker is Professor of Clinical Neuroscience at the University of Cambridge in the UK, and is a consultant neurologist at Addenbrooke's Hospital, where he sees patients daily. He is widely recognized as one of the world leaders in cell replacement therapies for Parkinson's, and he has been, as we all know, a central, long-standing figure in the development and clinical application of these therapies. As always, before we begin, please note that this session is for informational and educational purposes only. If you are seeking medical advice, this is not the place. Please consult a qualified healthcare professional. There will be time for questions, probably half an hour only. So we'll try to pick the questions that will be of interest to most of you. And the Q&A will, of course, be after the presentation. So if you would like to ask something, please use the Q&A function located at the bottom of your screen. Do not put your questions on chat, as I will not see them. Put them under Q&A, please. And for those joining us for the first time, No Silver Bullet is co-managed by Mark Lambert and myself. And our goal is to share Parkinson's expertise as we do tonight, and help our community make informed choices. We share all our webinars on YouTube and on Spotify, and you will find the relevant links in the chat at the bottom of your screen and on our website. The website is nosilverbullet4pd.com, nosilverbullet number 4pd.com. Now let's get started. Please join me in welcoming Professor Barker. Roger, the stage is yours.
Well, thank you very much. I hope my internet's going to be all right. I'm just going to put my slides up. And I hope that everyone can see that.
Perfect.
Perfect. Okay, thank you very much. Well, thank you very much for inviting me to talk on this topic, which is very apposite at the moment, given that there's been a lot of recent activity in it. So what I would do in this talk is I'll briefly give a rationale as to why you would consider using a dopamine cell therapy in Parkinson's. Then I'm going to talk a little bit about what sort of cells we could use, the history of using fetal dopamine cells, which I'll briefly cover as a prelude to talk about where we've got to with stem cell derived dopamine cells. And I guess if I start off by answering the question, are we close? The answer is we are close. There are multiple clinical trials ongoing with stem cells in Parkinson's, probably 15 trials, of which 3 have published. And major pharma is involved with it. So this is an area which is very active at the current time. So these are my disclosures. I obviously talked to quite a number of companies about this particular therapeutic, dopamine cell therapies. And I also spend quite a bit of time talking with other companies about other agents and patient-related outcomes, which is something I feel very passionate about. That's who funds me. And so let's begin. So obviously this is something which is very familiar to this audience about what is Parkinson's. But what's important for our discussion tonight is to understand a couple of things really about it. One is that the diagnosis is still in 2025 a clinical one. And so that does mean that everyone you see and put into trials, there is a possibility they may not have that condition. But classically, people present in a particular way. And that is related to pathology, which is extensive, not confined to the brain, involves the gut, but critically does involve a population of dopamine cells which sit at the top of the stem in the so-called midbrain in an area called the substantia nigra, because it's pigmented with melanin. And within that, you have about 500,000 dopamine cells that project up into an area called the striatum. And when you've lost half of those cells, so about 200 to 250,000 dopamine cells, you get the first clinical features of Parkinson's. And that's been known for many years. And has resulted in the treatments which exist for Parkinson's, which I know they vary around the world, but fundamentally the treatments for Parkinson's involve fiddling with this dopamine system. So you either replace dopamine in the form of L-DOPA, you stimulate the dopamine receptors with dopamine agonists, you stop the breakdown with various enzyme inhibitors. And over time, as many of you will know, whilst the drugs are extremely effective in the early years, they do lead to complications. And the commonest complication is that the drugs have variable effect. They sometimes don't work. They sometimes work too much, and you get dyskinesias. And that then leads to a sort of more advanced therapies, which starts with amantadine to deal with the dyskinesias, infusion therapies, deep brain stimulation, and more recently focused ultrasound. And the reason I say all this is for two reasons, really. One is to show that the core to treating Parkinson's and the effectiveness of what we have in clinic in 2025 is around the dopamine system, even though the pathology extends outside of that, and even though the symptomatology extends outside that. And secondly, because we have a lot of treatments, the success that this therapy is going to have to be very clear cut for it to be able to be better than what we have already. But for our discussion to begin with, what this sort of emphasizes is that there's a clear therapeutic target, the dopaminergic system. So why not simply try and rebuild it? Why not try and put back 250,000 dopamine cells on each side of the brain, and by so doing, put the system back to normal? And wouldn't that be a logical thing to do? So that is obviously what one's been thinking, and this is not a new idea. This has been around for over 50 years. And if you think, well, what dopamine cells could I use to repair that system, then essentially you can think of two particular sources. One could be from within. So can I find a dopamine cell source in situ in the brain, which I could recruit to repair this network that's failing and giving people the first features of Parkinson? So the first thing you could think to do, well, I've told you there are these dopamine cells in this area called the substantia nigra. You lose 250,000 of them, you get the first symptoms. Can't we just switch on the production of more dopamine cells in that part of the brain? We know that the adult human brain makes new nerve cells in certain areas to do with smell and certain memories. You make new nerve cells. Why can't we just switch on making more dopamine cells in the substantia nigra, which is a lovely idea, except that just doesn't happen. So there's no evidence that in the adult human midbrain, we make any new dopamine cells after we've been born. So the number you have when you're born is the number you will always have. So unfortunately, we can't switch on that system. An alternative approach has been, why don't we take cells where dopamine normally work, which is in the striatum, a bunch of cells called glial cells, which we have a lot of, and we're not quite sure exactly what they do, but could we borrow 250,000 of those glial cells in the striatum and directly reprogram them into dopamine cells? So take 250,000 glial cells, put in some factors into that bit of the brain and convert them into dopamine cells. And therefore you produce a new population of dopamine cells in the site where you want them using the patient's own cells. And that's a very exciting and very innovative area. But at the moment, the problem with that is the efficiency is very low. We can't get many to turn into dopamine cells, and we can't get them to turn into the dopamine cells that we want. And then the third approach, which is now in trials and has been in trials for a number of times, is to try and keep the dopamine cells you have alive, growing and sprouting. So can I actually preserve what's there and make them work more efficiently? And the way to do that is to give them some sort of fertilizer in the form of growth factors. And as many of you will know, a company called AskBio is actually doing a gene therapy currently with GDNF to try and regrow that pathway. So there are ways to think about replacing the dopamine cells using some internal source, but neurogenesis doesn't exist. Making new dopamine cells is exciting but inefficient, and growth factors are being trialed. But what I'm going to talk about for the rest of this talk is repair from without. So the idea here is to say, okay, the system's going to fail, it's going to die for whatever reason in Parkinson's. Let's simply put back 250,000 new dopamine cells, and we will get those dopamine cells from an external source, and we will transplant them into the brain. So that's the strategy. So then the question is, well, why bother when we've got so many other good, effective dopamine treatments that are out there? Well, I think there are three main advantages to this. The first is when you give dopamine drugs, they obviously are designed to replace the missing dopamine in the system that's failing. But what the drugs will do is they will stimulate any dopamine system that happens to be in your brain. And obviously you have a large number of dopamine systems, and some of those have little or no degeneration in Parkinson's. So when you give these drugs, they can stimulate things which they shouldn't do and give you off-target effects, of which probably the most common are neuropsychiatric problems, so driving hallucinations, confusion, gambling behaviors, and things of that nature. They can also exacerbate some of the other problems you see in Parkinson's around a thing called the autonomic nervous system. They make your blood pressure drop further. So if you could put dopamine back where it's needed and only where it's needed, you would avoid all these off-target problems with the dopamine drug. So it would be a cleaner way of treating the dopaminergic problems of Parkinson's. The second, which I've already alluded to, is when you give people L-DOPA medications, so Sinemet or Madapar in the UK, Stalevo and such like, all of these drugs, Stanic, then over time, those drugs, by not stimulating dopamine receptors in a normal physiological way, lead to the development of involuntary movements, these so-called L-DOPA-induced dyskinesias, which in the early stages aren't particularly troublesome, but over time can get worse. They can become quite significant and troublesome, and that leads to these other interventions. So if you could put dopamine back in the form of nerve cells, which could physiologically release dopamine at the site where you want it, you would avoid L-DOPA-induced dyskinesia. So you could stop off-target effects, you could stop the genesis of L-DOPA-induced dyskinesias, and it's a one-off treatment. I simply take you to theatre, pop in a couple of 100,000 new dopamine cells, thank you very much. And so what would that mean in terms of therapy for 2025? What would that actually look like? What is the vision? Well, in essence, if we could get this to work and we could get it to work consistently, we wouldn't be able to cure people with Parkinson's. But what we would do is your treatment would look like that. So we basically could obviate the need to take any of the drugs that you're taking and then all of the downstream complications that come from that. So this offers huge potential to radically change how we could treat Parkinson's around this dopamine system. So let's embark on that and say, well, what could we do in order to show that's competitive? So if we want to take this forward, we're going to look at dopamine cells to transplant in. What do we need to know to show that this therapy is really going to fulfill that vision, which I've laid out for you? So we have to show that whatever dopamine cell source we're using, that we can do it feasibly, so we can actually do it in the numbers that are needed, and it's safe. We have to be able to demonstrate that when we put the dopamine cells into the brain area we want, which is the putamen, the area of greatest dopamine loss in Parkinson's, we can put that dopamine system back to normal. So we can put the dopamine levels back to that which it was before people developed the condition. We need to show if we can do that, that the patients go back to near normal around the dopamine responsive elements of their condition, and that we can do this without producing side effects, which, as some of you will know, when we use human fetal dopamine cells, which I briefly mentioned, create side effects of graft-induced dyskinesias. These are the involuntary movements which I talked about with L-DOPA, which of course I can always get rid of because I can always get rid of L-DOPA. But in this case, they were driven not by L-DOPA, but by cells within the transplant. And obviously, once I've transplanted the cells, I can't get them back. So for any cell therapy, dopamine cell therapy, we have to show we can do it feasibly, we can do it safely, we can put the bit of brain back to normal in terms of dopamine levels. That makes patient near normal, and we can do that without producing side effects. So what cells could we use? So there are two sources, and the first source that was really considered was back in the 1980s, led by the team of Anders Björklund in Lund and his clinical colleague Ulla Limbaugh. And they argued that actually what would be a useful source of dopamine cells to treat people with Parkinson's would be the developing dopamine cells in the midbrain of human fetuses. Now, that obviously creates some ethical problems because in order to collect those cells, you need to collect them from termination of pregnancies, so women who are having abortions. So what would happen is the woman decides to terminate the pregnancy, that decision is made, the tissue is then collected by an independent team who then dissect out the developing dopamine cells from the midbrain of that aborted fetus and transplant it into the brain. That is the source of the dopamine cells, ethically very contentious, but for our discussion this evening, it's been very useful in a proof of concept. So the idea here is I'm going to replace the lost dopamine cells in the Parkinson's brain with a new population of young developing dopamine cells from the human fetal brain. So this went on for many years. So these trials have been done over many years, and I'll come on to the latest trial that we just finished. And what it demonstrated was that in certain cases, the benefits were extremely impressive. So this is two cases that I'm briefly going to mention from London, who had Parkinson's disease for 10 years at the point at which they traveled to Sweden, in Lund in Sweden, where they had fetal dopamine cells transplanted into their brain. They were then followed for the next 20 years. So on this graph, you can see where they had the first and second graft in the patient on the left. It's not labeled on the right in patient number 2, which is B. What you can see is they had UPDRS is a scale of the disability you have with Parkinson's. The higher the score, the worse the Parkinson's. And you see if we just concentrate on patient number 1 in figure 1A, the patient had a response after the first transplant. They had another response after the second transplant. They were done in sequence, one side of the brain and then the other. And then after about 5 years, the patient had shown a marked improvement. That score that they now had was equivalent to what they had when they presented to their neurologist 15 years previously. And then they remained extremely stable for the next 15 years. So 30 years into their Parkinson's, they have a UPDRS motor score, which is the same as when they presented, and they've stopped all their medication. And on PET imaging, the area that's been grafted has gone back to normal. So this demonstrates in certain individuals, you can get long-term clinical benefit to the extent of decades of improvement with the patient stopping the medication. There was another patient in Sweden, a Swedish patient who had a transplant on only one side of their brain. They had a similar response. They improved, they came off their medication, but then sadly went on to develop a Parkinson's dementia, which makes the point that these therapies are not curing people. And 24 years after those cells were put in and the patient died, they acquired the brain and they looked at it. And everything you can see on this brown here are the transplanted cells. They only had the transplant on one side of their brain. So this demonstrates that a quarter of a century after you put these fetal dopamine cells in, they can still be found, they're surviving, and they innervate the brain. So the Parkinson's brain may be putting some pathology into the transplant, but it's not killing it. So this shows you can get long-term clinical benefit with long-term survival. Now, as many of you will know, in the 1990s, this then moved to the States, where they had 2 randomized controlled trials where patients either had a transplant or sham surgery. The upshot of that was that actually none of the patients as a whole showed an improvement. And for the first time, they were seeing side effects, which had not been seen in these early studies, of patients developing these involuntary movements. So by 2003, it was generally felt, as DBS, deep brain stimulation, was becoming very popular, that the day of dopamine cell therapies had come and gone. It didn't really work when you put it to formal trials, even though it was clear that individuals, as I've shown you, showed a marked clinical response with restoration of dopamine levels back to normal, with post-mortem evidence to support survival of the cells long term. So an alternative explanation was it can work, and when it works, it's transformative, but it's not working very consistently. So the question is, how could we make it more consistent? So we embarked on a meta-analysis where we tried to collect all of the data from all of the trials around the world to then analyze to see what were the results across the globe in people who had undertaken these studies. So these are the major studies that we got the data from. And the details aren't too important, but what's interesting is that this is their scores, their change in their motor scores 2 years after they've had the transplant when they aren't taking any medication. So in the defined off period, they haven't had any medication overnight, 2 years after they've had a transplant, this group in the red box are those who've got a 33% more improvement, greater improvement in their Parkinson's, which is more than you would expect. So 2 years after they've had the intervention, they're 33% better in the off period when they're not taking medication. So then the question is, well, what characterizes those patients? What makes them different? And what it looked like was that these were younger, less advanced patients. They were also patients who had received more tissue. So they'd received more fetal tissue, not 1 or 2 fetuses, but the dopamine cells derived from 3, 4 or more. But these were patients who had a longer duration of immunosuppressant. So more aggressive immunotherapy seemed to also support a better clinical response. And I'll come on to talk about longer follow-up in a minute. But this meta-analysis led to us apply for a new trial to look at human fetal dopamine cells in a population of younger, less advanced patients using more tissue preparation and a more standard immunotherapy regime. So this was called Transneuro, and it has 2 arms to it. One was this observational study where we said, we think this group, who we've defined through our meta-analysis, will be the best group for receiving these transplants. And that's exactly what we found. This is a complicated graph, but essentially what we found when we follow people over a period of time, in this case 3 years, this group of younger onset, less advanced patients have a very linear progression. So they look as though they're going in a very standard way where any intervention would be clear to see. The ACE at the bottom is cognition. There is no evidence that they have any problems with thinking, and they don't develop any dyskinesias. So on the background of collecting this group of patients, we then randomly selected some of these patients to have transplants of fetal dopamine cells. And although the original study was to graft 20 patients and then do a bigger study, ultimately we could only graft 11 patients during the transeuro period, which extended from 2010, with the last follow-up, last patient in this study being in 2021. So it took 11 years to graft 11 patients. And so this is what we found. This was the paper that appeared earlier this year. And essentially what we found was we could not put people back to normal, but we clearly saw something. So this is all a bit complicated. I don't know if you can see my pointer here, but essentially this is a control group. So the blue line are people who we've just followed up in our standard follow-up every 6 months from 4 years before they had a transplant to 3 years after in the grafted group, but the control group had nothing. And as I've already shown, they show a beautiful linear deterioration over time. The top group, which is the transplant group, they were a little more advanced, but at the point at which we did the transplant, subsequently, they slightly flattened off. So clearly there was a point of inflection when we put the dopamine cells into their brain. So the dopamine transplants had done something, but clearly it hadn't returned people back to normal, because if you remember earlier in the patients who had done very well, 20 was the sort of level which patients had dropped down to. So we didn't see that they had improved back to normal. And when we did dopamine scanning of their brain, this is the group that had no transplant. This is the putamen, the area of greatest dopamine loss. This is the normal range up here. In people with Parkinson's, it's obviously low. And if you don't do any transplants, then it gets worse, as you would expect. In the transplant group, they didn't get worse, but they didn't go back to normal, but they got a bit better. And on a second dopamine scan, we saw exactly the same. So there was a partial clinical response. There was a sort of stabilization of dopamine loss in their brain. And then on 2 other measures, the amount of dopamine drug they needed to take was less in the grafted group, and the amount of time they spent with the drugs not working in the off period was also reduced. So all of this would imply that the patients had been partially treated. And to show this more graphically, this is a transplant PET scan. So this up here is the caudate, the dots at the top of the caudate, and then you should have a tail down here, which is the putamen. In the patient who had a very good response, so this patient went back to normal, you can see that this is before the transplant. After, they got massive red blobs there. So we put dopamine back to normal there. But you can also see in this bottom patient here that we haven't seen that response. So it was a very uneven response, but overall the group looked as though there had been a partial response. In other words, we'd underdosed them. So we hadn't given them enough dopamine cells, and that some of the patients developed some side effects. Now, why did we not put more dopamine cells in? And why did we only graft 11 patients over 11 years? Well, the problem was, in order to transplant one side of the brain, you need to collect dopamine cells from 3 fetuses. And for every patient we managed to do that, we had another 4 collections that failed. So every patient had a 1 in 5 chance of ever having a transplant. So in the course of the transeuro transplant study, we did 21 transplants, 10 bilateral and 1 on one side. Each was a separate surgery. So for the 21 surgeries we did, we had a further 87 surgeries that were cancelled because we had insufficient tissue. So to come back to those questions I raised at the beginning using human fetal dopamine cells, it's clearly shown a proof of principle. Is it feasible and safe? Well, the answer is no, it's not feasible. We simply cannot use that. Regardless of what the results in transeuro have been, we cannot use that as a source of cells. It does seem to be relatively safe, and it can be extremely effective, but we can't do it consistently. So in some people, we put dopamine levels back to normal, but in the majority, we do not. However, when we do put it back to normal, the patient goes back to normal. So the principle is there. And can we do it without producing side effects? No, we couldn't. Even in our trial, where we tried to be much more selective about what we did, we still had problems with these graft-induced dyskinesias. So whilst human fetal dopamine cells have been very informative and shown a proof of principle, if this field was ever to move forward, we had to find a better source of dopamine cells. And so that comes to the possibility of using human pluripotent stem cells, of which there are two types. So there are embryonic stem cells, which come from the spare embryos in IVF programs. So all of us at one point in our life was an embryonic stem cell. So essentially you have a spare embryo from an IVF program, you take the embryonic stem cells, you turn it into a stem cell line, you make dopamine cells from it, and you transplant it into the brain. That was first done with human embryonic stem cells by Jamie Thompson in 1998. The alternative is to take adult cells, so take any of your skin cells or blood cells, reprogram them into stem cells, so-called induced pluripotent stem cells, and then turn them into dopamine cells and transplant them into the brain. And that became a reality in 2007 when Shinya Yamanaka in Kyoto managed to do that with human cells. So in the last 30 years, we've suddenly had the possibility of using human stem cells to make dopamine cells. And when we grafted our first patient in TransEuro in 2015, the amount of money going into stem cell therapies for Parkinson's disease by major biotech and pharma was approximately $0. By the time we grafted our last patient in 2018, that had grown to several billion. Why? I have no idea. But now this field is incredibly active. So these are the ongoing trials that are taking place in Parkinson's disease using a stem cell-derived dopamine source. So Aspen Neuroscience in California are using autologous. They're using the patient's own cells, turning them into dopamine cells and transplanting them in. At Mass General, they've done one patient, which they published in 2020. They've now got a company and an academic group are doing autologous iPS cell transplants. Arizona State University has just completed a trial, I think, of induced pluripotent stem cell-derived dopamine cells in people with Parkinson's disease. Blue Rock in New York, which I'll come on to in a minute, have published a trial and now in a phase 3 study. We've just completed our trial. There are 3 trials in Japan, in China. Japan have completed a trial and South Korea have completed a trial, and both of those have been published. So here are the published trials that have taken place. One was in the New England Journal published in 2020. I'm not going to talk about that, but I am going to talk about these other 3 which have appeared in the last 6 months. So the first is this study from New York. The second is this study from Kyoto in Japan. And the third is from Seoul in South Korea. They're all small numbers, 12, 7, and 12. And I'll take you through each one and how I see the data in terms of what it's telling us. So this is the first one which I want to talk about, which is the Blue Rock study. Blue Rock now are part of Bayer. This was a company set up by Lorenz Studer and Vivian Tavar. And so this is a study where they took human embryonic stem cells, so cells from a spare embryo. This is a very well-established line. They then have a protocol where they can turn those embryonic stem cells into dopamine precursors in 16 days. They then put them into vials, freeze them, tested them in animals to show they were safe, and they worked. And then they did a study where they took 5 patients and injected in both sides of the brain at one point a low dose of cells, so 1 million cells into each side of the brain. And then they took 7 patients where they put in 3 times the dose in one operation onto both sides of the brain. They published their data at 18 months, and the patients were immunosuppressed with standard immunotherapy for organ transplantation for a year after the grafting. So this is the data. So in the low dose group, obviously, if you're going down, you're getting better. You can see that there's a bit of variability, but essentially over the 18 months, you can see that in the low dose group, there was some improvement in the patients. However, in the high dose group, there was a much more significant improvement. So their mean score before they had the intervention was 50, and 18 months after they had the transplant, it had halved to 25. So that's quite a significant clinical improvement. If you look at their PET data, which is extremely difficult to do on this graph, Unfortunately, the PET data didn't show any massive improvement in dopamine levels in the brain. So on the scanning, it was less impressive than the clinical response. And also, over the course of the 18 months, there was no reduction significantly in the amount of medication the patients took. So whilst there's been a clinical response, unlike in Transuro, where the patients did have a bit of an improvement, there was a bit of an improvement on their scan. They clearly reduced their medication needs, and some of the other measures were better. Here we can see a clinical improvement without a major change in imaging, certainly not back to normal, and no reduction in their medication. So I would say that this trial showed that it was safe. There were no major side effects that emerged. There were some clinical effects at the higher dose, but quite what was mediating that was a little unclear from the data that we have. However, on the basis of that, BlueRock were invited by the FDA to have rapid transfer to the clinic. I can't remember quite what it is. Accelerated registration, that's what it is. And so they have now embarked on a phase 3 study of 102 patients, of which it will be 2-to-1 in favor of the transplant. So of the 102 patients, 2/3 of them will have a transplant, a third will have imitation surgery, so have a hole drilled in their skull but no cells put in, and then they will compare the outcome. And if that outcome is positive, then the company will be able to apply to offer that therapy to people with Parkinson's. So that is a phase 3 study which is already underway. The second study I want to talk about, so that scientific paper was published in Nature in the summer. This second study, which was published in the same issue of Nature, is from the group in Japan, led by Jun Takahashi. This was a slightly different study, but the same principle. Here they took human-induced pluripotent stem cells. They froze them down into the freezer. Then they took an aliquot of this, and for every patient, they then took the iPS cells, ran it through a differentiation protocol to make the dopamine precursors. They again showed it was safe and worked in their animal models, including non-human primates. And then in a smaller study, they then grafted in a single operation 3 patients at low dose and 4 patients at a high dose. They then followed them for 2 years clinically with a series of imaging, and again used immunotherapy, although in this case it was less aggressive, just 1 drug over a 12-month period. And what they showed, so this is their score. So again, just to orientate you, the higher you are, the worse they are, the lower you are, the better they are. So this is this change in your motor score when you've not taken the medication overnight. And this is the percentage changes over here. So this is obviously before the operation, and then this is 2 years after. So there is an effect. You have to look at this slightly cautiously, because in animal models, these dopamine cells take at least 6 months to have an effect. So they take 6 months to turn into dopamine cells, connect with the host brain, and have an effect. So if you move the y-axis 6 months to the right, which is when the transplant should be working, you can see that the results perhaps are not quite as impressive as with baseline. So there may be an effect there, but it's obviously not as striking as one might initially have thought. However, their imaging is quite impressive. So whilst they have not put dopamine levels back to normal, you can see in this bottom scan, here is the caudate, here is the putamen. You can see over a 2-year period, the red blobs emerging of the transplanted cells. Again, in this cohort, it's only 7 patients, there was no reduction in their dopamine medication. There were again no major safety issues, but again, the outcomes were a bit variable. But there was, I think, more evidence of graft survival, even though the clinical response was perhaps not as striking in this case. In Japan, as you might know, they have a different way of taking advanced therapies forward. You can apply to have a 7-year license to offer your therapy to patients on the back of a study like this. All patients can then have it. At the end of 7 years, you have to then be able to demonstrate that those patients in that 7 year that have had your therapy have shown a benefit. If they have not, the license is removed and you can't offer it anymore. The company that Jun and his team work with, Sumitomo, have also now planning to do a trial of this cell therapy in America. And then the third of these studies, which is from South Korea, which was published in the journal in Cell the month before last, so about 6 to 7 weeks ago. They used an embryonic stem cell just as the group in blue in New York had. They essentially did the same approach. They took embryonic stem cells, converted them into dopamine cells, grafted them into patients. They had 6 patients at a low dose, 6 at a high dose, and they published their 1-year data, although the study's still ongoing. Again, what you see here is that there is a clinical response, both at low and high dose here. So the patients do improve. Again, I haven't got the absolute numbers, but that's about 60 dropping to about 50. And in the high dose, it's from 60 to about the same 45. And if you look again at this plot on B, you can see that. But if you use this 6-month cutoff, then perhaps again, the effect is not as striking as you might have seen. But there was nevertheless evidence on the scans of some cell survival. Again, it was perhaps less impressive than in the Japanese group. And just for comparison, this is the high dose group down the bottom here. This is the caudate. And you can see that there is a slight signal there, but this is our very good patient in the Trans-Euro study. So it's not as marked as we've seen in our best patient using fetal dopamine cells, but nevertheless, it shows it's safe. There is some clinical effect, and there was some, again, evidence of graft survival on PET imaging. So these are just the trials that are published. These are all the ongoing trials which I've already mentioned. So there is a lot of activity going on around these at the moment. Some of these have already submitted their papers. Some are still ongoing. Our own study, STEM-PD, is a collaboration between myself as the clinical lead in Cambridge and Marlin Palmer, a brilliant stem cell biologist in Lund, who's the overall coordinator of this study. We're essentially transplanting embryonic stem cell precursors into patients with moderately advanced Parkinson's. For those who think this is relatively straightforward to do, this is just a little bit of the sequence of events you have to go through in order to get permission to do that. So that's all the various meetings we had over a 5-year period just to get to the point where we could apply for a clinical trial. We got approval in Sweden, then we got approval in the UK, and then all of this was done with Novo. And as some of you will know, Novo Nordisk have over the summer had a new chief executive who has decided to close down all of their cell therapy work, not just in Parkinson's, but in diabetes and heart for business reasons, nothing to do with anything else. So that is currently getting us to think a little bit at the moment. We grafted our first patient in February of 2023. We had a staggered process where we grafted people sequentially, 4 at 1 dose, 4 at a high dose. Last patient, last visit was in October of this year. So we're just in the process of writing up on that. So to conclude on the stem cells, using the same principles as I did for the fetal tissue, if we ask those same 4 questions, is it feasible and safe to treat people with Parkinson's using human pluripotent stem cell dopamine transplants? Yes, it is feasible, but it's not without risks. So none of the studies have reported major risks. We had a major problem with immunotherapy in one of our patients. Can we put dopamine levels back to normal consistently using stem cell derived dopamine cells? The answer is no. But nevertheless, they've moved to phase 3 studies. Can we see people go back to normal with dopamine transplants from human stem cells, which put dopamine levels back to normal? No, not really, because we can't put dopamine levels back to normal in the striatum. And does this actually work without causing side effects? And the answer to that is yes, in that we've not seen any of these dyskinesias, which are a feature of the fetal dopamine cells. So the positive data is that yes, it's feasible. It does seem to give some signal of efficacy, but nobody so far has managed to restore someone back to normal. It may be early days, either on imaging or clinically. So that brings me to the final slide, which is really about the challenges. And I would say the problem here is that we're still in the early stages. So we still do not know how to deliver these cells. We don't have a device for it. There is no device that is on the market for delivering cells. And we certainly don't know the dose of cells to be given. So we don't actually know how many cells to give, because only the minority survive grafting and turn into dopamine cells. So this, I think, is a major problem. And all of the trials, I would argue, have underdosed their patients, which is why we've only seen partial responses either clinically or on the imaging. The immunosuppression is clearly something that we need to consider. And when I talk to one of the patients in our study, as I do on various webinars, he will say that the biggest problem he had in the year was the burden that the immunotherapy exerted on him, not the surgery, and not a worry about the cells. And not that the immunotherapy caused him side effects, it was just the increased number of tablets and some of the lethargy that induced. Ultimately, for these therapies to be successful, they're going to have to compete with what else is out there. And as I've said, there's a lot of very good therapies that work for the dopaminergic aspects of Parkinson's disease. So we've still got a long way to go to show that these therapies can really do that. And then ultimately, where do we go next with these type of therapies? The beauty of stem cells is we can engineer them to do all sorts of things. We can engineer them to get rid of the aspects of them that generate an immune response. We can engineer them to get rid of the protein that could be causing Parkinson. So the transplants are completely immune from the disease process forever. So there are lots of opportunities there. So finally, I just want to thank all the people who are involved with this for our own trials, both Transuro and STEM PD. Huge numbers of people were involved. We were enormously grateful to Novo Nordisk Cell Therapy R&D team, all of whom are now redundant as of the last 3 months by Novo. But without them, we couldn't have got to where we've got to today. And then finally, all of the funders who've helped our work, much of which I haven't discussed, the wonderful collaborators I have, but particularly the team in Lund, where we've had a very rich and wonderful collaboration for nigh on 20 years. Marlin Palmer, Agneta Kirkeby, Anders Bjorklund, and many others there. And with that, I'll finish, and I'm very happy to take any questions.
Roger, thank you very much for this presentation. I think that we're not there yet, but I think that everyone on this webinar will have now a much clearer understanding of where we are and the various teams in the world, the way they're progressing and the results they're achieving. So thank you for this. I just wanted to ask you perhaps to say a word of caution for people who would see adverts on Instagram about solutions in Mexico or Switzerland. I don't want to pick on any country, but I think that this is not really matching what we have heard tonight. So what are the risks, and should people stay away from that?
Yeah, thanks, Michelle. I mean, there is a difference between stem cell therapies of the type I'm talking about. These are basically groups that have worked for years developing a dopamine transplant or dopamine cell from a stem cell source, and they're part of clinical trials. There are lots of clinics which are out there offering you a stem cell therapy for Parkinson's disease. Those stem cells are often of very different sources. They're not going to turn into dopamine cells. You have to pay for them. And this stem cell tourism is a bit of an issue, really, because all trials at the moment with dopamine cells are experimental. And as with any experimental therapy, if you are paying for it, that should not be the case. People should be reimbursing you for your travel. You should be fully recompensed for being in a study. You shouldn't be paid for being in a study, but actually all trials should be formally funded, and you should therefore just be in the trial without having to pay for it. And it is a big worry, and I do warn people against it, because there have been, when people have had these therapies, not so much in Parkinson's, but there's certainly a patient with spinal cord injury had a stem cell therapy. One of these clinics developed a spinal cord tumor. There was a child who had a rare neurological condition, had a stem cell therapy, developed multiple tumors and died. So these treatments are dangerous. So I would really tell anybody, if they're interested in this, the ISSCR has a very good site on this, but only join up to studies which are fully funded and which you're being recruited to into a formal trial.
Thank you very much. Let's go through the questions. We have roughly half an hour, so we have a big audience tonight with close to 300 people. I will not be able to, Roger Barker will not be able to, with my help, answer all the questions, but we will try to pick the most relevant ones. John was actually the first question we had today, and he was asking whether stem cells treatment only improve motor symptoms, or is it meant to improve all symptoms, including non-motor?
Yeah, good question. So basically, these are not cures, as I keep trying to make. They basically will be a better way of taking dopamine, is the fundamental message. So what improves with dopamine is what will improve with these cell therapies. So they're primarily designed to treat motor symptoms, so that in particular, the slowness and stiffness. Now, that having been said, if you can sleep overnight because you don't have any off problems, your sleep will improve. If your sleep will improve, so will your daytime fatigue. so will your cognition. There's also some thinking deficits which happen because of a loss of dopamine in the striatum where these cells are put back. So what it will help with is it will help primarily the motor, some of the non-motor, which are a secondary effect of having a better motor control, probably some cognitive effects, and then it will avoid some of the problems you get in Parkinson's because of the medication that you take around somnolence and autonomic. So it's not designed to treat the non-motor. That may come in the future.
Thank you very much. We have two questions here about genetics. Someone is saying that anonymously he or she has a PINK1 mutation, while Edward mentions that he's asking about GBA1 mutations.
Yeah.
So what is the impact of, I know that 85%, I think, of all Parkinson's cases should be coming more from the environment. But for people who have like a genetic profile, such as the PINK1 or the GBA1, does stem cell therapy is supposed to work as well?
Well, there's no reason it wouldn't work, because in either case, you've got a loss of the dopaminergic nigrostriatal pathway. So repairing it would work regardless of the etiology. Yes. Now, obviously, some people have thought about Parkin, actually, because Parkin is a rare form of Parkinson's. It's very pure dopamine loss. So that might be a good condition to treat. The problem with Parkin, which is a good thing, is it progresses very slowly. So PINK1, I would say no, there's no problem. GBA, I wouldn't say there's a problem. There's always a worry with GBA because there's this story about it presenting earlier in a more aggressive fashion and people developing a lot of non-motor problems earlier in the disease course. So there may be problems there that if you restore the dopamine system, you won't stop some of the other, non-motor features from developing. Yes. And the other thing to mention is, of course, I'm only talking about stem cell therapies here. There's a lot of interest in genetic forms of Parkinson's for gene therapy and for other therapies around specific problems. So GBA, there's at least 3 gene therapy trials going on around GBA. PINK1 I know less about.
Thank you very much. Rich and Paula are asking actually, and it's really very much to the point of you showing that map of the world with all the efforts worldwide, what you personally have learned from trials happening in other parts of the world, what have been the main couple of things that are really for you, that moment of the little bulb lighting up in the room, that some new concept has been discovered or some new avenue has been opened?
Well, one of the things we did very early on was in 2014, we decided, so Marlon and myself decided that actually it'd be very helpful if the main groups in the world met every year to discuss their progress. So we met with the group in Japan and we met with the group in New York. So the Blue Rock group, or in those days it was the group run by Vivian and Lorenz, and the group in Japan. And we would have a 1 or 2 day meeting where we would discuss all the data we had unpublished and where we were going with our clinical trials. And the idea was to try and harmonize what we did as much as possible, whilst allowing people to obviously develop their own programs of work. So we tried to learn as we went along, so it wasn't seen as a race of one against the others, but we try as a concerted effort to deliver the best possible therapy to as many people as possible by taking a very considered and collegiate view to it. That still carries on. It obviously becomes trickier once companies get involved because they're less keen for us to be able to share all that data. But I think what's been very rewarding in this field is people are very happy to share what they've got and to try and learn from each other. So I think we're learning all the time. And one of the things I always like to point out is people always get a bit disappointed, especially with my recent talks on this, that it hasn't made people, dramatically better. But these are early days. I always say Christian Barnard, people will remember him of a certain vintage, that he did the first heart transplant. The patient who received that heart transplant, I think, lived for 10 days. So it made no difference to them whether they had a heart transplant. But actually, you learned something vital about that, which was you could plumb it in and you could get it to work. The question was, how could you stop rejection? So every trial tells us something. A good trial will always give you useful information to work on. So I think we're trying as much as we can with the community to progress at a rate which tells us what we know and what we don't know, and be honest about that. It is a little tricky now because companies obviously are looking for a return on their investment. So there is things, I would say, moving a little ahead of where the data is in some places.
Yes. Thank you for this, Roger. Dietrich is asking, his question is really about the neuronal cells. How do we know they will produce the required amount of L-DOPA? And actually, I think this is probably still an open topic because you said that some of the other regions had used injections that were perhaps not strong enough to achieve the results that you want to see. So is there a dose question? How many?
So the problem is trying to guess the dose. So what we want is obviously we want about 200,000 dopamine cells to survive in the Parkinsonian brain when we've grafted them. So how do we guess what 200,000 is? Well, only the minority of the cells we transplant turn into that. So basically in a rat, in a model, we will inject, say, let's just say for the sake of argument, 100,000 dopamine cells, and 10% of those turn into dopamine cells. So 100,000 into the young rat brain, we get 10,000 dopamine cells. We say that's a great result. So then we say, well, in order to get 200,000 in the human brain, we need to put in 2 million. Now, obviously, that's a guess, because we're going to guess that the number that turn into dopamine cells in the aging Parkinsonian brain will be the same as in a young rat brain. The delivery device we use in the human brain is obviously different to a rat. The immunotherapy we give in people, we don't give to rats because we use immunodeficient rats. So there are all sorts of differences between them. And so we're guessing really what that dose is. And I think the problem is that the devices we use and in the brain where we're injecting them, it's quite hostile for cells. And so less of them are surviving than we anticipated. So trying to get the dose right is going to be critical. And until we've done that, I think these big trials run the risk of being a bit premature.
I understand. There are a few questions about China. There is a specific question here about first affiliated hospital of the USTC in China. Apparently, they have announced over the weekend that 6 patients showed rapid symptom improvement. I don't know if that was something that was on your radar screen.
Yeah. So I think what you have to be a bit cautious of is rapid responses. Because as I tried to demonstrate with the fetal dopamine cells, the maximum benefits were seen at 5 years. And why does it take so long? Because what you're putting in is you're not putting in a dopamine nerve cell. You're injecting a cell which is destined to become a dopamine nerve cell. And in rats, that takes 6 months. So if I inject these cells into a rat brain, I see no change in the animal's behavior for 4 to 6 months. Then I start to see an improvement. And that's because the cells have to go in, they have to survive. They then have to carry on and turn into what they're supposed to come into, dopamine cells. They then have to make projections. They then have to connect. And then you have to learn how to use that, and it has to mature. So that takes months to years. So if patients are improving within the first few months, 6 months, for example, then it's hard to believe that it's the dopamine cells that are doing it. And so the worry, of course, with all of this is how much of it is a placebo effect? How much is it investigator bias? And that's why I think ultimately, to see how effective these are, you have to wait to see what the long-term benefits, and do they correlate with changes in dopamine on scanning and dopamine requirements that in terms of medication.
Still on China, Iman is asking whether the Chinese studies have the same gates, he's using that word, as people have to go through in the USA. So in other words, do they have the same regulatory kind of like framework, or are they able to move faster with potentially some downsides, of course?
Yeah, I don't know the answer to that, because I don't know what the regulatory framework is in China. It does vary, or did vary, between state hospitals and military hospitals as to who could do. So they have different ethical processes for those two. Different countries have different regulations, and that can be quite a problem. So Japan, for example, has this very unique way of licensing drugs for 7 years, or therapy for 7 years. You can't do sham surgery in Japan. That's just not something they will regulate. In the US, the FDA won't allow you to do a trial currently with these without ultimately doing sham surgery. So different countries have different perspectives on things. So it's always difficult to judge. The key thing is that what people need to put in their papers is an honest account of the origin of the cells, the permission to use it, the ethics for doing the trial, and then what the consent is that the patients agreed to be involved with that.
So David is asking here about the cost of the treatment. I guess it's very difficult to answer specifically, but do you have a perception that if Bayer, Blue Rock, for instance, after the phase 3 trial, decide to, with the FDA approval, commercialize this, do you think this will be something that is affordable, or will it always be a Rolls-Royce only available for the very few?
Yeah, I don't know the answer to that. What I can say is that the manufacturers, I think the reason this field took off, apart from the intuitive sort of attraction of putting in dopamine cells for a disease that has a dopamine loss, is the manufacturing. So in our hands, if you start with a 6-well plate, so a little like with 6 little wells of embryonic stem cells today, then by Christmas I would, at Christmas Day, I would have made enough dopamine cells from that 6-well plate to treat 300 patients. So that's just me.
Yes.
So I can have one plate, I can make enough cells to treat 300 patients in 16 days. If I had a robot, 1,000 plates, I could treat 300,000 people. I could treat the world, basically, with that one manufacturing run. So the manufacturing run is relatively short. It's a standardized product. So actually, it should not cost a lot of money, because the cost is often the production run and variability. The shorter the production run, more consistent, and the less time it costs to actually manufacture. So I don't think that ultimately it'll be that expensive, but that's my view, and I'm not going to set the price.
No, I know. Dan picked up on the fact that you said that in the early trials, the subjects presented 20 years later, just as they were when they began the trial. So he's asking whether actually what we did is stop the progression, or did we really roll back the symptoms? If they were in the same state as at the start of the depression.
So I say what we do is the disease continued. So we didn't stop the disease, but what we did, the disease in the nigrostriatal pathway, this dopamine pathway, has essentially been bypassed because we've just rebuilt it. So the disease carries on, but our transplant is replaced and repaired that bit of the network, and is basically, you put it back to normal.
Yes. Thank you. Ashley is basically thanking you for the webinar, and he's a medical student, very passionate about Parkinson's. We love you, Ashley. Thank you very much. He basically is asking whether you think that some of the studies have simply not have had the length of time to produce results that were valid to interpret.
Well, so that is a good point. And obviously, they're normally published for two reasons. So a lot of the studies have already published, and that's important that they're published soon to show really about feasibility and safety. So we're writing up our 12-month paper. We're not writing up our 12-month paper on the expectation we can show whether it works or not. We want to show what our safety profile looks and can we do it. So people need to know that information. I think what will be very interesting is the 3 to 5 year follow-up on these patients to show what actually the clinical benefit is over that period of time.
Exactly. So Dan lives in Grand Rapids, and he heard you speak at the Grand Challenges conference a year ago. So he's thanking you for having presented there, and he's asking a couple of questions. The first one is that is the Van Andel Institute involved in any of the research that we talked about today? And the second one is that do you expect more trials in the US?
So I don't know whether the Van Andel is involved, I'm afraid, because I don't know. I mean, it's more of a research institute, the Van Andel, rather than a clinical institute, but obviously they're linked to the local clinical services. So I don't know. Chicago's just up the road. They've obviously got a very big Parkinson's research set up there. There are a lot of trials going on. So in the US, I know of at least, I think 5 or 6, 7 trials going on. So these are likely to become much more commonplace. And obviously for the Blue Rock Bio study, that's 102 patients. I know they've gone to Australia. I don't know where else they've gone for their studies. They've obviously been in Canada, and that study which I mentioned, that was Canada, that was Toronto, and the study which took place. So there are clearly centers, they're looking for people to be suitable for it. I think what's important to understand is that for a lot of these trials, the real frustration for the patients is that there's very strict inclusion-exclusion criteria, and people get a bit cheesed off if they say, well, I'm 73, and they say, well, upper limit 72, you can't have it. And you say, yeah, but I'm a terrific 73-year-old. So unfortunately, these are the criteria. But yeah, there are a lot of activity at the moment. So I would definitely keep a lookout for trials, because there will be a lot of activity in this area.
Thank you very much. Someone is asking about Novo Nordisk strategic decision, and actually, presumably, I don't want to presume. They're asking if you're looking for another partner at the moment.
Well, Novo Nordisk are looking for a partner to partner with our program. Essentially, we did all of the Marlin did all of, and Agneta did all of the preclinical work. Our trial, which we've done, STEM-PD, we got a grant from Novo Nordisk, but it's an academic study. They hold the IP and the cells and such like. So they're looking for a partner, and we're optimistic that they will find a partner so we can carry on.
It will be exciting. I wish you all the best. Randall is asking whether the therapy we're talking about today can be utilized in conjunction with DBS.
Yeah, it's a good question. So obviously, if it works very well, you wouldn't need DBS. So that's what we're after. At the moment for trials, you can't have a stem cell trial therapy if you've got DBS, just because you've had previous surgery and we're not quite sure how difficult it would be to do surgery, even if it's different target sites. Also, there's a question whether the regulators would say, would deep brain stimulation alter your stem cell therapy? On the other hand, if you have a stem cell therapy and then you need DBS, that wouldn't stop you having DBS. So the two are not mutually exclusive. It's just at the moment there are issues around whether you can recruit people into trials with DBS.
Yes, understand. Charlie is asking quite a good question, actually a bit long. I will read it to you. He basically says that although levodopa just boosts dopamine neurons produce other neurotransmitters, for instance, GABA. And he's asking whether the transplanted stem cells dopamine neurons produce other transmitters. And does this mean that stem cell therapy will provide wider benefits than just replacing levodopa?
Yeah, so there's a lot in that. So, I mean, obviously the simple approach is that we're just trying to get dopamine cells to produce dopamine, and because we know dopamine works. Most of the cells in the transplant are not dopamine cells, and quite what they are, we don't know. One of the worries with the fetal tissue was that where the dopamine cells develop in the normal brain, next door to it, there are a group of cells which produce serotonin, 5-HT neurons. And it's thought that those cells mishandle dopamine, which was what was driving the graft-induced dyskinesias. We know with our stem cell preparations, we get no serotonin, no 5-HT neurons there at all. So that's why we don't think we'll get that side effect with it. Whether the transplants produce other things which might be useful, we don't know. It might not just be transmitters. They may be producing, levels of growth factors, antioxidants, other things which may be useful. But at the moment, we're only concentrating on whether they can make enough dopamine. But the point is well taken, because Agneta Kirkeby, a good colleague of mine in Copenhagen, we've worked on the idea that we could not just replace dopamine cells, but cholinergic cells, a different transmitter, acetylcholine. They sit at the base of the brain, they project up, and they go wrong in Parkinson's and drive some of the thinking problems. So could we take the same strategy using stem cells, engineer them into cholinergic cells, and graft them in? And then you could do both.
That would be amazing. Yeah. I have two questions about age, really, or I would say almost stage in life. I have, first of all, sorry, let me just get to that. Torben Jaeger is asking, what would happen if the stem cells would be transplanted sooner in the progression of PD, when the patient still can engage in Parkinson's modifying behavior? Yeah. And I will let you know the second one, actually, is I think you mentioned that the therapy could be more effective in younger patients. I don't know if that's right, but actually, what are the characteristics of a patient who is a perfect candidate? So what happens if you are early in your condition, and is there a perfect candidate?
Yeah, well, I don't know. We don't know if there's a perfect candidate. I mean, the meta-analysis we did with the fetal dopamine cells was to try and identify who seemed to have the best response. So it seemed to be younger patients with less advanced disease. Quite why that is, I don't know. It may be that the receptors and all of the problems which go wrong with long-term L-DOPA have been less damaged, if you like, by the medication, and therefore they're easier to integrate dopamine cells into that network. I think younger patients have a slightly more, plastic vein, so they're better able to cope, with the surgery. They probably have a slightly more friendly environment for the cells, and they can integrate more. I mean, my ambition would be, if this works, then you would go early in the disease. I mean, the problem at the moment is, of course, no ethics committee is going to say, Roger, go and transplant these newly diagnosed Parkinson's patients. And they'll say, what do you think could happen? I said, I have no idea. And he said, well, what could you do? Well, I've got about 20 drugs I could give them. They'll do crackerjack with that. So, the ethics at the moment restricts it. But ultimately, if it works, there's no reason you couldn't have this as an early stage therapy to stop you taking the drugs, to stop you developing the problem.
Exactly. I think we'll keep going for another 10 minutes, and that will be for tonight. I still have quite a few questions, but we're doing well. Or you doing well? Jeff is asking a very basic question here, but at the same time, I don't think I could provide the right answer myself. What is the difference between dopamine replacement versus gene therapy?
Okay, well, so gene therapy is where you inject a gene into the brain, and that gene carries something that you want it to make. So, for example, we were involved with 2 trials. First one was with Oxford Biomedica, and the second was with Axovant, where we had a gene therapy, or they had a gene therapy, and the gene was injected. It went into cells, and what it had attached to it were the enzymes to make dopamine. So you inject it in, you infect the cells are infected with all of the structure needed to make dopamine. So when you gave someone a drug, they could make dopamine in cells that didn't normally make dopamine. You could put a gene therapy in with GDNF, which is currently in trial at the moment. The idea there is you inject it into the brain, the cells take up the gene, they then have the gene for GDNF and they produce GDNF. So there are ways of delivering. That involves infecting cells in the brain to turn them into producing something you want. What we're doing here is we're giving people a new population of dopamine cells. And obviously your drugs work by being swallowed, taken up through your gut, taken up into your brain, then taken up into nerve cells and converted into dopamine in the cells with any site, but mainly in the site where you need it.
Thank you. Kiki is asking actually whether the people who were doing well across the trials had some common characteristics.
Yeah. Well, no, I mean, all you can say is, coming back to that meta-analysis, they tended to be younger, less advanced patients. I think, it is a big challenge to know who the optimal patient is and whether there is somebody who's optimal. The problem is, especially with the fetal tissue, is, of course, it wasn't a standardized therapy. So, if the three of us on this, Michelle, myself, and Mark were having a transplant of fetal tissue, we would all have to collect the tissue from a collection of fetuses. We would all have to have those so it wouldn't be the same in all of us. So there are differences in the cells as much as there are in the patients, which makes it a bit hard. Stem cells are more standardized. So I still don't think we know is the answer.
Someone is asking about the adverse effect, the adverse events. You mentioned, of course, dyskinesia arising from this treatment, but aside from that, I had read an article about someone who had like psychosis and went through a very long period of adaptation. I think in Lund, if I remember well. But actually, what other side effects have you seen?
Yeah, so basically, I think the side effects are obviously, if we do it in order, there's the surgical risks. So obviously, you're having implants, so you could have a hemorrhage, you could have stroke. So far, we've not seen any major neurosurgical complications. There's the complications from people with moderately advanced Parkinson's coming into hospital for surgery. And so one of the problems in the Lund patient, which I think you mentioned, was that they had become a little confused whilst in hospital, which is not uncommon, especially if you've had a long anesthetic. These operations take, 8, 12 hours, so they're not quick, so they're long procedures. The transplant itself shouldn't produce a psychosis. I mean, obviously, there's a worry that it produced too much dopamine, but that's never been seen ever in any patient. They produce too much dopamine overall. So there's the risk of the surgery. There's the risk of the Parkinson's misbehaving because of the surgery. There's a chance of the Parkinson's getting a bit worse because of the immunotherapy. There's the risk of the immunotherapy in terms of infections, bone loss and things of that nature, which one tries to deal with by giving prophylactic antibiotics and bone protection. There's a worry, obviously, that the cells could turn into a tumor, but, Marlin, for example, has done over 2,000 transplants of these cells and never seen a tumor. So that seems extremely unlikely. There's a worry that the cells would migrate to other sites, but we have no evidence for that. So the major side effects, I would say, are a disappointment that they're not going to help, the risk of the surgery and the risk of the immunotherapy, but the cells themselves are probably safe.
Thank you very much. I think I have an interesting question here, tongue-in-cheek, by Tim, who basically says the title of the presentation is "How Close Are We?" Now, while, of course, he wasn't expecting you to give us a specific date in the future as to when this would be available to patients, what would be your comments as to your estimate? And he says, this is tongue-in-cheek, he will donate £100 to cure Parkinson's if your answer does not refer to 5 years. Which I have heard.
It's always 5 years, 5 years' time. Well, the reality is that Blue Rock and Bayer are in a phase 3 study. And I don't quite know when the endpoint of that is, but that presumably because they've grafted their first patient in September of this year. So I imagine that trial will be done in 3 years. And if that trial is positive, then that will be a therapy. I mean, obviously there are hurdles to go after that, but in theory, if that's a positive trial, then there's no reason the therapy couldn't be licensed.
Exactly. I think I will leave it at that. We have a few questions left, but actually a lot of them are very specific to those persons' specific situation. I think that actually we have covered a lot of ground, and I don't want to keep you so late that you will refuse to talk to us next time in 5 years.
Yeah. Well, now you're going to have me back in 3 years' time to see whether I was
That's right.
close to getting it right.
But thank you very much, not only for making yourself available today, but for making yourself available to helping us with our condition every day of the year. So Roger, thank you so much for your time. It's much appreciated. And before I let our audience go, I just wanted to say that on the 12th of January, we will be delighted to welcome back Dr. Simon Stott, who you know very well. Simon is Director of Research at Cure Parkinson's, and he will review the key Parkinson's research takeaways from this year, 2025, and tell us what to watch for in 2026. So until then, on behalf of No Silver Bullet, I'd like to wish everyone a very happy Christmas or festive season and a great new year full of health and happiness, I think is the other thing I would wish to everyone. So Roger, thank you so much. Thank you to our audience. See you next time.
Thank you very much. Bye-bye now.
Bye-bye.